Yeah, he's almost starting. Yeah, ready? Okay, we are ready, Eric.
Okay. Okay,
So
perfect.
first, I'm going to introduce you. Okay. So Professor Erik Ullian, he's a professor from the ophthalmology department at the from the School of Medicine from the University of California in San Francisco. He graduated in biological science at the University of Chicago, and he did his PhD at the UCSF in neuroscience and a postdoc at the Ben Barry's lab at the Stanford University. He's, uh, he's a director of the Vision P30 Core grant over the past 5 years and previously served as a co-director from the Computer IT Core from over 10 years. Actually, his lab is exploring the role of glial cells in supporting and regulating visual systems in visual function. He has more than 50 papers at PubMed, a bunch of talks in very recognized scientific conferences, and I'm very happy because I have a student at his lab now. So thank you, Eric, for that.
Of
And
course.
thank you for accepting to give this lecture in our discipline today. And the floor is yours.
Oh, great. Thank you. Thanks so much for the introduction. So, today I'm going to be talking about sort of two streams of work we're doing, trying to model complex disease in both the cortex and the eye using various types of organoids. And before I go into the details, let me just see if I can switch slides here. Before I go into the details, of course, I just want to acknowledge the people who actually do the work. I just write the grants. And so I've had a lot of very talented people over the years in the lab, including Martita DeMajo, Mark Kuntz, Nir Salinas, um, Marcus Lee, and Kriti Chaplot, who've been in the lab. And I've collaborated with a bunch of very good collaborators at UCSF, including Michael Ward and Martin Kampman and Eric Wong, who have helped us tremendously in our studies of neurodegeneration. So what motivated us to start looking at neurodegenerative disease was actually the original work we were doing in neurodevelopment. So we started off as neurodevelopmental biologists, and we, along with a bunch of other people from the Allen Institute, from Ben Barris's lab, Michael Olden's lab, had discovered this interesting observation that if you compare the gene expression of neurons and glial cells, for example, between mouse and human, there's quite good conservation of gene expression, even between mouse and human neurons. But when you start looking at glial cells, such as astrocytes, oligodendrocytes, and also endothelial cells, you see that there's a much greater divergence of gene expression between humans and even to some extent uh, from humans to non-human primates, but certainly from human to mouse. And so there's been some speculation that glial cells, astrocytes, oligodendrocytes, et cetera, microglia, may actually have been under more evolutionary pressure as the brain expanded and cognitive function changed and so forth. And so one question that we began to ask is whether there may be fundamental differences in the glial cells between human and mouse that may drive some of the observations of the types of diseases that humans are susceptible to that mice are not very susceptible to. And of course, there's been increasing findings of the interaction between astrocytes, microglia, and neurons driving all kinds of both neurodevelopmental and neurodegenerative diseases. I know that many people in the audience, I believe, are probably interested in autism spectrum disorders, and there's increasing evidence for microglia playing a role in those types of disorders as well, not just neurodegenerative diseases. And so, one question that obviously comes up is how do you model this in a system? How do you model human astrocytes, human microglia, and human neurons in an organoid-like system? And one of the challenges with conventional organoids is that when you grow conventional cortical organoids, the timeline of astrocyte generation is very slow. So neurons are obviously first born, and then astrocytes and other glial types. And so you need to culture the organoids for, on the order of a year in order to be able to get those astrocytes. So we tried a different approach, basically, which is to differentiate the cells separately, either using transcription factor induced expression or even conventional differentiation, which takes a long time, but you can expand and bank the cells so that you have tens of millions of cells. And then with those cells, put them back into single cell suspension and then aggregate them into these assembled organoids. And the advantage of this, in our view, is that it allows us to have very tight control over the types of cells that are in each organoid. So they're all very repeatable from organoid to organoid to organoid. We can really have tight control over the ratio of glial cells to neurons, which we think is important and certainly is different in different brain regions in human, different between human and other non-human species. And we can also incorporate microglia into these organoids very easily. And so just, as an example of what we see, for example, with isogenic wild type and granulin knockout cells, which I'll focus on for the first half of the talk, we can start to see pathology that is seen in human brains, such as TDP43 leaving the nucleus and forming these ring-like structures. We can see that pathology in these 3D organoids, and not in isogenic controls that are cultured for similar amounts of time. So, what is happening in these 3D organoids? So, of course, the first thing we did before even looking at the neurodegeneration phenotypes is we looked at the astrocytes. And one thing that we noticed in these 3D organoids, so these are images of astrocytes. These are GFP labeled astrocytes in the 3D organoid. And this is over a period of a couple weeks where they start off as small but highly process-bearing cells and then rapidly turn into these extremely large cells that are very good matches for the endogenous large astrocytes that are seen in the human brain, and have many of the features of endogenous human astrocytes. So you guys may be familiar with the work from Naked Nadergaard and other groups that show that human astrocytes have these very unique features that are not found in other species. They're found to some extent in non-human primates, but even humans have distinct types of astrocytes. So they're distinguished by their size. They're very large, and they tile in a very specific way. And we can reproduce a lot of those observations in these 3D organoids, in addition to changes in gene expression. So, and also you guys should feel interrupted anytime if you have questions about what I'm talking about, because we don't have to go through all slides and I can skip slides as well. So given that, we at least feel like we have a good model for human astrocytes, is this going to now allow us to study astrocyte neuron interactions, astrocyte microglia interactions, and so forth? And we think it will, and this is an example of a paper that's on bioarchives that we're trying to finally get published in a journal with Martin Kampmann. And what this is an example of CRISPR screens that we've done in neurons that are cultured either in these 3D assembled organoids compared to the same cells cultured in more conventional 2D. And looking for changes in gene expression and changes in survival of the neurons in the 3D environment versus 2D environment. And there's several interesting observations that we've made in these 3D organoids. So the first is if you look at a UMAP, you can see that the cells over the course of a couple of weeks, they move towards these trajectories of maturation. So the cells become mature very quickly in these 3D organoids. And this is also illustrated here, where we compare these neurons, these NGN2-induced neurons in conventional 2D cultures, for example, at 2 weeks, versus the 3D cultures at 2 weeks. And you can see that there's this big increase in these sort of classic markers of mature neurons. Many of these are synaptic markers, and they come up very early and they get maintained in these neurons in the 3D organoids. And we find that not only are the neurons becoming more mature, but also the astrocytes and the microglia are showing increased markers of maturation. So they're becoming more matched to endogenous human astrocytes and microglia and neurons in these 3D cultures than if you take the same cells and you plate them down even together in a 2D culture. So there's something about this 3D assembled organoid environment that allows this type of maturation to occur. And of course, some of these, I know, again, there's some people in the audience interested in autism spectrum disorders. certainly some of these genes have been linked now to autism. And so I think in terms of studying, synaptic mechanisms that may underlie certain types of autism spectrum disorders, I think this type of approach could be very informative in terms of what's going on. When we look at actually the survival dependence of these neurons in the 3D culture compared to the same neurons again in the 2D culture, we also see this enhanced dependence of these neurons for survival on genes and pathways that are known to be involved in neurodegeneration. So 2D neurons, 2D human neurons in culture, they're relatively insensitive to perturbations of some of these sort of classic neurodegenerative genes, surprisingly, in the 2D culture. And that may just be because they have essentially an infinite supply of glucose and goodies from the media above them. But in the 3D culture, they show a survival dependence on these well-known disease pathways. So again, we think that this is one potential advantage of this type of assembled 3D organoid, is that we can uncover, as the neurons become more mature, we can uncover these pathways that play critical roles in normal brain survival, normal neuronal survival. And we know that, for example, there's increased coupling of lipids and metabolites between the astrocytes and the neurons, and the microglia and the astrocytes and the neurons. So we think this cross-coupling of metabolism and the cell-cell signaling is really what's driving forward some of this change in survival dependence. So given that, one of the first questions we asked is whether we can use this assembled 3D organoid to model neurodegenerative diseases, which have been so classically challenging to model both in mouse and in conventional organoids. And as I mentioned before, a particular class of diseases that we were interested in is frontotemporal dementia, which is, thought to sort of be broadly similar to other types of neurodegenerations that occur, like in Alzheimer's disease. But frontotemporal dementia is characterized by typically a very specific neurodegeneration that occurs both in the frontal and temporal cortices, and it's characterized by this loss of this nuclear protein TDP-43 from neurons, and it becomes extracellular. And the reason, of course, that one of the reasons that we were very interested in going after FTD in particular is because there's now very good genetic understanding of what some of the drivers are of this neurodegeneration. And this just shows some of the genes that drive frontotemporal dementia. And I'm going to be mostly talking about granulin mutations, but we've also now been looking at C9orf72 mutations and are seeing very similar types of phenotypes and optineurin and TBK1 as well. And so for granulin, which I'll be talking about, all the disease mechanisms seem to be due to loss of function. So loss of granulin expression in the brain. So there's actually a childhood leukodystrophy that's a homozygous deletion of granulin, which is a very severe early neurodegenerative disease. Most people who have granulin mutations have a single mutation in an allele, and it's a much more slowly progressing disease under those conditions, where it typically hits in the early 50s to early 60s. So one question that we always think about as developmental biologists is what aspects of any phenotype we see are cell autonomous versus non-cell autonomous. And so the first question that we asked when we had these isogenic granulin knockout neurons, or iPSC cells, and controls, is what phenotypes, if any, do we see in the neurons themselves? So are the neurons sufficient to show phenotypes? Do we even need to bring in any of these other cell types? And this just shows some examples of these neurons that we have in conventional 2D culture, stained for progranulin. So these are the wild type, isogenic wild type lines. And you can see progranulin is localized right near the nucleus in these cells. And these are actually lysosomal structures. So granulin plays really critical roles in the lysosome. It seems to have a lot of other roles as well, which is a little bit challenging when you're trying to understand exactly what's happening in these cells. But it clearly plays very important lysosomal roles. So we know it co-localizes with the lysosome. Here's the isogenic cell that only has granulin, both alleles of granulin knocked out. And in this case, we just see no progranulin staining in these neurons. So they seem to be devoid of progranulin. And interestingly, much to our surprise, when we look, for example, at TDP43 in the wild type or isogenic granulin knockout cells, we actually can't detect any quantifiable difference in the amount or localization of TDP43 in the neurons alone. So the neurons seem to not really be bothered too much by the loss of this gene, which we know at least leads both to FTD, and when you have a homozygous knockout, leads to this very early leukodystrophy and severe neurodegenerative disease. So, being a glial biologist, of course, the next question we ask is, what about astrocytes? What do we see in the granulin knockout astrocytes? And so here's an example of the isogenic lines made into astrocytes, wild type in the top row and granulin knockout in the bottom row. And you can immediately see that there's differences in the morphology of these cells. So this is what, healthy, happy human astrocytes, even in 2D, look like. They're highly process-bearing cells. They're GFAP positive. But the granulin knockout cells, actually, the astrocytes in particular, have much more of this sort of, classic reactive type phenotype where they look more fibroblast-like. We see increased expression of ataxin in these cells, and they just appear to be under more stress, both by gene expression and immunostaining. And interestingly, if we do, for example, a classic phagocytosis assay on the astrocytes, either using, Frodo zymosin beads or synaptic purified synaptosomes, we see this great reduction in the ability of these granulin knockout astrocytes to phagocytose material. And it's actually even more severe for synaptosomes. So they seem to have an even greater deficit in their ability to engulf synapses compared to these synaptosomes. And when we look at lysosomes and compare lysosomal changes in the neurons or the astrocytes, we also see these consistent differences. So as I mentioned, granulin is known to play really important roles in the lysosome, although what exactly it's doing and how is not mechanistically fully understood. And in wild type neurons, you see these beautiful LAMP1 positive lysosomes right next to the nucleus. These are also granulin positive. In the knockout, the lysosome is actually largely intact and pretty normal, but we do actually see a very subtle phenotype of these sort of beads of lysosome that start to move down some of the processes, like the dendrites and the axons. But it's pretty subtle, and the neurons themselves seem to have fairly normal lysosomal function when we look at it. In contrast, when we look at the astrocytes, so here's a wild type astrocyte, they have a lot of lysosomes in them. Astrocytes have a lot of lysosomal activity because they have a lot of membrane, and they're turning over membrane and proteins quite rapidly compared to neurons. But in the granulin knockout, we begin to see these large lysosomal structures that appear sort of frozen in the cell. And when we look for so for example, we see this increase in lipid droplets, which is good because that's described in the leukodystrophy, this increased lipid droplets. So we can recapitulate that in vitro. And it actually looks, based on various experiments that we've done that I'm not really going to have time to go into all the details, it looks like the whole sort of endosomal to lysosomal pathway eventually just becomes frozen in these astrocytes, and they're no longer able to effectively phagocytose things like synaptosomes and move things to the lysosome. They basically just get stuck in the absence of this granulin protein. All right, so neurons, subtle phenotypes, astrocytes, much more severe phenotypes. What happens when we put these two together? Can the astrocytes actually do something to the neurons and make them sicker? And I think you guys already know what the answer to that is. And so just to tell you how we initially did these experiments, we again made these assembled organoids where we made the astrocytes and the neurons and assembled them in these large microwell plates, like 96 well plates, where we have uniform size and ratios of cells. And then, we can slice them or do biochemistry on them and assay them. And this just shows sort of low power image of TDP43, which is this important nuclear protein that's commonly mislocalized in neurodegenerative diseases, in the wild type neurons in the top row and the isogenic granulin knockout neurons in the bottom row. And you can hopefully see immediately that there seems to be some changes occurring in the TDP-43 that you can see expressed in the granulin knockouts and where it is. You can see that these are cell bodies here, and here you can see many fewer cell bodies, even though there's the same number of cells at this point, and much less localization of the granulin within the DAPI positive cellular nucleus. And interestingly, when we look in higher magnification of cells, such as neurons, and I'll show you pictures of neurons next, you can see that the TDP-43 protein starts to form this very characteristic ring-like structure, and you can even see it starting to go down pieces of dendrite in the neurons. And these are the types of observations that have been seen in human TDP-43 postmortem tissue. So we think, at least on a basic cellular level, we're able to recapitulate some of the cell biology that's occurring in TDP-43. And this is extremely hard to recapitulate in other systems. So you don't see it in mouse. It's even hard to recapitulate in 2D cultures where there's TDP-43 pathology. These types of ring-like structures are very hard to see. People can detect loss of TDP-43 sometimes in severe cases of disease in neurons, but not necessarily these types of aggregations. And this just shows that this is actually happening in the neurons. So here is MAP2 in green, and this is a neuron, and the granulin knockout now on top and the wild type on the bottom. And you can see that there's actually loss of this TDP-43 in these neurons that are here. It's not only occurring in neurons. We do occasionally see it in other cell types as well, but it is very prevalent within the neurons, just as it is in the human cortex. And this is just quantifying these data, showing that in the wild type cells with astrocytes and neurons, we see this what we call volume of occupancy when we measure the intensity of TDP-43 within the nucleus. And in the granulin knockout, we see this highly significant shift to a lower nuclear localization of TDP-43. And interestingly, we do also see, as I mentioned, we saw this ataxin stress granule marker in astrocytes. We do also see an increase in ataxin in all cell types in these 3D organoids, coincident with the change in TDP-43 localization. And ataxin doesn't seem to be a cell autonomous cell autonomous issue if you culture these neurons on their own, but seems to be induced in some way by the astrocytes in these neurons. Of course, we want to know when the phenotypes occur. So the images I showed you were about 30 days, about a month of culturing in these 3D organoids, which, an organoid world is pretty fast. Interestingly, if we look at 2 weeks, we don't see any, at least by the methods we use, any clear statistically significant change in the localization of TDP-43 in the neurons or astrocytes, although we do actually see an increase in ataxin already at 2 weeks prior to the change in TDP-43. So this is also, I think, a fairly important point, because it suggests that unlike other manipulations that have been done, like directly targeting TDP-43 and knocking it down, this is a spontaneously developing over time phenomenon induced non-cell autonomously in neurons that leads to phenotypes similar to the very slow progression of loss of TDP-43 and pathology that occurs in human brain. And so, obviously it's much faster in these 3D organoids than it is in the human brain. But at least I think at some cell biological level, we are able to recapitulate some similar phenomena that are occurring in these 3D assembled organoids and the brain. And this is just remind you what the human brain looks like with these clear ring-like structures, and you see these TDP-43 starting to go down the processes, very similar to what we have observed. So there's been, in the last 5 years, there's been all these realizations about what loss of TDP-43 actually leads to in neurons, and primarily, people haven't looked as carefully in other cell types like astrocytes and oligodendrocytes, although we're very interested in that as well. And TDP-43 is this very interesting nuclear protein that plays a really important role in preventing missplicing of its target mRNAs. So there's many mRNAs that have these what are called cryptic exons or introns in them. And TDP-43 basically prevents those from being included when the mRNA is spliced, so that you get a normal spliced mRNA and you don't get the inclusion of these weird introns within the mRNA. And so in the absence of TDP-43, when it leaves the nucleus, you start to get these misspliced mRNAs. You often get truncated mRNAs that include these cryptic exons that should be removed, but aren't removed. So they're treated more like introns. And then you can detect that by qPCR or by doing single cell RNA-seq now. And so this is, again, just to remind you, the phenotype that we see with physical loss of TDP-43. Here's a paper from Prudencio's group showing in human brains, where they looked at large populations of human postmortem tissue and controls in FTD that had TDP-43 pathology, and another type of neurodegeneration that did not have TDP-43 pathology. They saw this increase in cryptic missplicing of target mRNAs, in this case, a stathmin-2 mRNA, which is a very robust readout of TDP-43 pathology. And in our 3D assembled organoids, we can totally recapitulate this. We can see a clear increase in the misspliced stathmin-2 mRNA. We've now repeated this many, many times. It's even been repeated in other labs now. So it's a very robust phenomenon that's not just unique to our lab, but to other labs who are working on these cells as well and using the system. And this is true not only for stathmin-2 mRNA, but other target mRNAs that have been identified. So there's been now a whole family of mRNAs that have been identified as targets of TDP-43. And we can recapitulate the cryptic missplicing of those target mRNAs in most cases, where we at least have good primers to detect them. So we're at least recapitulating this important aspect of the human pathology. In addition, this is, again, results from Prudencio's lab showing this increase in phospho-TDP-43 in patients with these FTD TDP-43 pathologies. We see by biochemistry clear increases in phospho-TDP-43 in these 3D assembled organoids, not in the 2D cells, but in the 3D cells. And we can even detect it by immunostaining to some extent. So again, another classic feature of human neurodegenerative disease that we can recapitulate in these 3D assembled organoids. And finally, I think this is perhaps most biologically significant, is in patients where TDP-43 leaves the nucleus and starts to aggregate, there's this well-characterized fragmentation of TDP-43, where TDP-43 basically gets cleaved into these smaller forms of TDP, so TDP-25 and TDP-35. Now, TDP-35 is less prevalent in the human population, but it does occur, and TDP-25 is quite prevalent in the human population, as is extremely difficult to reproduce in vitro and doesn't occur in mouse. And we can actually detect both isoforms. So if we look at TDP-35 and TDP-25 here, which is a little bit faint, but you can see it's higher here than in the controls, only in the insoluble fraction and only detected with the C-terminal antibody, not the N-terminal antibody, which is exactly what you see in the human brain. We can detect very clear and significant increases in TDP-25 and TDP-35. So I think that this is potentially really informative about what is actually happening to TDP-43 in these 3D assembled organoids, and suggests that some of the biological processes that lead to fragmentation of TDP-43 and aggregation that occur in the human brain, at least on a cellular level, are occurring within these systems as well. And so hopefully this will allow us to gain more biological insight into the biochemistry, basically, of TDP-43 pathology. So, going back to our interest in astrocytes, I showed you this evidence that astrocytes have a stronger phenotype, neurons have a weaker phenotype. And I sort of implied that maybe astrocytes were driving the phenotype in the neurons. But to really test that, one of the advantages of this 3D assembled organoid that we can take advantage of is that we can mix and match cell types. So we can, change who has the mutation and who doesn't, especially when using isogenic lines. So we can culture 3D assembled organoids and define numbers and ratios of cells, but in this case, maybe only with the astrocytes with the granulin mutation and wild type neurons, or only the neurons with the granulin mutation and wild type astrocytes. And we can ask, okay, in what case do we see TDP-43 pathology? Which cell type may be the main driver? And so when we do that experiment, we find this very interesting observation. So when the astrocytes have normal granulin gene expression, even when the neurons have no granulin in them, we see pretty normal cryptic stamen 2, suggesting that there's not a lot of TDP-43 pathology. And this is confirmed by looking by immunostaining in the top. In contrast, if the astrocytes are lacking granulin, but even if the neurons contain granulin, we actually can detect clear increases in cryptic stamen 2, as well as some evidence of these ring-like structures and loss of TDP-43 from the nucleus of neurons. It's not as severe as in the case where both cell types are missing granulin, but it's significantly different from wild type and a clear trend. So this has been published by Martina Demajo in this paper. And so this really does support the idea that the astrocytes are the driver for at least some of the TDP-43 pathology in the case of the granulin mutation. And we're actively exploring other potential forms of neurodegenerative disease where we think glial cells, astrocytes, and microglia may drive some phenotypes in neurons, and neurons may be relatively less sensitive to loss of gene function than the glial cells are. And finally, I just want to show you that we can rescue this phenotype. So, here's an example of the astrocytes and neurons wild type with very little cryptic stamen 2. Here's an example with both cell types missing it with an increase in cryptic stamen 2. And then if we add the granulin protein back to these cells extracellularly, you can see, first of all, that it gets back into the cells. So here's the knockout cells after we've added the protein back. It goes into the cells and pretty much localizes where we think it should, and it actually rescues the cryptic missplicing that we see. So we're pretty confident because of that what we're seeing is actually directly due to the loss of function of this gene that's been linked to TDP-43 pathology and neurodegeneration. So basically, this is just a summary of what I've shown you. Even in these assembled organoids of just astrocytes and neurons, we can start to sort of pull out these interesting observations and cellular processes that we think are very relevant to neurodegenerative diseases, such as the loss of TDP-43 from the nucleus, the phosphorylation of TDP-43, the increase in cryptic missplicing of target mRNAs, and fragmentation of TDP-43, all phenomena that occur in human neurodegeneration but are extremely difficult to recapitulate in other systems. And this is just with the astrocytes and neurons. And we know, for example, that the microglia are severely affected by loss of granulin, just like the astrocytes are. They have very similar changes in lysosomal structure, in ability to phagocytose, in their expression of various cytokines and other things that in theory should be detrimental to neurons. And in this 3D system, we can make the microglia quite easily from iPSCs. And they incorporate really well into these 3D assembled organoids. And here's actually an image we were able to catch of a microglia, translocating into a 3D organoid. And here's the, earlier picture from Cajal showing macrophages translocating through endothelial walls and blood vessels. And so once they go in, they start to extend processes and they sort of populate this area. And so we're actively pursuing this. And our preliminary data suggests that the microglia may be exacerbating the phenotype. So the phenotypes may become even more pronounced in the presence of these mutant microglia. And so we're hoping, by looking at all these cell types individually and together, we can really start to pull out the cell autonomy versus non-cell autonomy of some of these neurodegenerative diseases and understand how these genes are affecting neurons, which are the things that we ultimately want to save, but through these other types of glial cells. So That's all I have for that. And then I was going to switch to talking about uh, eye organoids. So if there's any questions, I can stop and answer questions right now before I make the sort of cognitive leap to retinal organoids.
Hi, Eric. Can you hear me?
Yeah, I can hear you.
Okay. So does anyone has any questions about this first part? Well, yeah. I have someone here.
Hi, Eric. It's working?
I
Hi, Eric. How are you? Thank you very much for an amazing talk. Actually, my postdoc is watching online and she has a question for you. Let me read here. In traditional organoid formation protocols, the self-organization of cellular localization within the organoid structure is a key biological feature of these models. If there a self-organization factor influencing the cellular arrangement and positioning within these assembloid structures? If so, how relevant is it, and would it be available to investigate further?
Yeah, so that's a great question. So one of the disadvantages of these 3D assembled organoids is that we can't get a lamination-like structure that occurs in the cortex in cortical organoids, where there's some lamination, although it's not perfect lamination. However, the astrocytes and the neurons do self-organize. So we do very little other than dissociate them and put them together and define numbers and ratios. And then these cell types know what to do, basically. They know how to arrange themselves so that there's neurons and astrocytes near each other, and they form this beautiful intercalated set of cells that are highly process bearing and in very close proximity to each other and sort of providing metabolic support for each other. So in that cellular aspect of self-organization, we can actually recapitulate that to a large extent. And in some cases, I'd say even better than you can in a conventional organoid, because you don't have continuous generation of new neurons and new astrocytes coming in and starting to develop. Basically, everything develops at once, and then you have these highly complex cells interacting with each other. But obviously, we can't capture, at least in this iteration of assembled organoids, we are thinking about how to do this. We can't capture the types of lamination and cellular diversity that occurs. So in this case, these NGN2 neurons are primarily a single type of neuron, which may be beneficial for some of the cell biology we're trying to do, as a more simplified cell biology system, because all the neurons basically behave the same. So we can get very clear statistical separation of these phenotypes that we're looking at. It wouldn't be surprising if we were able to make the system more complex and have some layering in it as well, which we think we might be able to do, that some of these phenomena might be harder to see at a whole organoid level, but maybe easier to see at an individual cell specific type level. So we may have to change from just grinding up organoids and running qPCR to doing single cell RNA-seq in order to really detect and understand disease mechanisms. But that would be an ultimate place to go, because we'd really be interested in understanding some of the cell type specificity of these diseases, which hit different types of neurons more than others, even in the cortex, even in frontal cortex. Upper layer neurons seem to be more affected than lower layer neurons, and these NGN2 neurons luckily are more similar to upper layer neurons than lower layer neurons. But yeah, it's a really important question.
Can you hear me then?
Yeah.
Okay. Uh, so great talk, Eric. Amazing results, and I'm very happy to see that we can. It's one more example that we can recapitulate things that we see in someone's brain using our organoids, in vitro organoids. It's amazing. So because of I have some questions, but I can write you later because of the time, and then you can go to your second part.
Okay, so this is going to be short. I'm not going to
Yeah.
talk about this very long. I just wanted to really throw this in as a contrast. So in contrast to these cortical organoids, where we can sort of make things separately, dissociate them, put them together, at least recapitulate some nice aspects, we think, of the cell biology that's occurring in the, in the target organ. For the eye, it's been extremely difficult to develop these same techniques to study eye development and eye neurodegeneration. And part of the reason we think is the complexity of eye development, where you have these strange placodes that form, and all this complex signaling that occurs between the external part of the eye and the retinal part of the eye that are required for normal development. So we've had a really hard time using transcription factor induced differentiation of various retinal cell types, and especially of photoreceptors. So for eye development, we're still going through those sort of classic, very long-term classic organoid, more like a cortical organoid, in order to be able to develop these organoids to the place where we can get photoreceptors and start to study photoreceptor degeneration. But just to show you under these conditions where we can culture these for a very long time, we can actually get organoids that are able to develop some of these features very, very rapidly. And so we're trying transcription factor methods in order to express transcription factors and see if we can make, for example, retinal ganglion cells to study optic neuropathies and glaucoma. And we're trying other methods to make photoreceptors. But so far, it's been extremely difficult to get that to work consistently. And so this is basically what the conventional organoid system looks like that we're using, where you start with 2D iPSCs, you turn them into embryoid bodies, you plate them down under conditions that promote retinal signaling with FGF and other factors, and then you start to form these outer segments that are actually retinal radioglial cells. And then over the course of a very long time, 100 to 200 plus days, you can actually see this fuzz that forms on the outside of these retinal organoids. And that fuzz is actually the outer segment of the photoreceptors, which we're very interested in. I'll just show you. So this is just details of how they're made. And you can see these beautiful, beautiful pieces of retina basically that are forming here. But one thing that we're interested in is trying, and also they layer beautifully. So in relation to your question about cortical organoids, just like cortical organoids layer nicely, in retinal organoids, you can actually capture that layering, I think, even a little better than you can in cortical organoids. And that's, I think, partly because of the smaller structure of the retinal organoids compared to cortical organoids, which tend to grow very big. We've certainly grown them in the past. But these retinal organoids stay small, and you can get really nice layering and separation of cell types, ganglion cell types and interneuron cell types, and then the beginnings of photoreceptor cell types. So I think there's a lot of potential for people interested in vision for some early circuit development questions, even using these retinal organoids. And one question that we have is whether we could use these retinal organoids with thalamic organoids. So this is a thalamic organoid and retinal organoid, and whether we can use them at relatively early time points when the retinal ganglion cells are there to get innervation of the thalamus and potential target innervation into the appropriate target. And the reason I'm interested in this is that in my past life in the mouse, we've done a lot of work on retinal ganglion cell heterogeneity and targeting and trying to understand some of the basic signals that drive retinal ganglion cells to target very specific types of neurons or laminations of neurons in their target area. And we want to see if we can recapitulate that to any extent in a human system. And so these are early days, but we actually can. So here's the retinal organoid, here's the thalamic organoid. I apologize for the not great pictures, but you can actually see when we start to pull them apart, you can see that there are axons actually coursing from the retina into the thalamic organoid. And we have much better tools now for this. So we have things like halorhodopsin MAPT lines where the axons are bright red and live neurons that we're using to really try to understand targeting and see to what extent we can recapitulate the reasonably well-defined targeting that has been characterized in mouse and macaque in human and actually macaque iPSC systems. So I just wanted to throw that out there. I'm not going to really talk about that anymore because I know we're out of time, and this is really ongoing work. Um, but um, but it's, you know, it's a question that we're hoping we can make some progress back to sort of our original interest in developmental biology, and try to use these human systems to ask questions about specificity of targets and circuits.
It is stuck it? You finished?
Yeah. I'm done. Yeah.
Awesome. So thank you, Erik. It's amazing. I remember that I saw this picture when I was in your lab, this last one with the neurons. Making the connections. Amazing picture.
Yeah. So we're
Yes, this
excited by
one.
that.
Yeah, that's very, very nice. So thank you very much for your talk. It was very interesting, and it's amazing job. And I'm asking. Yeah, we have questions here.
Erik, I'm just curious about your staining when you use LAMP1. Is a robust expression of this protein in your knockout cells. And I'm just curious if you staining as well for multivesicular bodies, because is multivesicular bodies is also acidic compartments. And I saw distribution, widely distribution in your cells. And I'm, wondering if you also check the extracellular vesicles secretion or something like that.
Yeah, these are really important questions. And I wish I was a lysosomal biologist after getting into this granulin work, because they're incredibly important questions. And I'm trying to collaborate with people who can basically help us answer those questions, because my expertise is not in lysosomal biology. And I get asked very similar questions to what you just asked a lot when I present this. And there's clearly something happening to lysosomes. We know that there's something happening, and we have some preliminary evidence that things are being secreted that shouldn't be secreted, or released through some pathway that we don't quite understand from the astrocytes, for example, and probably from the microglia, that may impact neurons very negatively. And so this is incredibly important, and we're trying to understand the methods to address this. So if you can direct me to some good papers on this as well, and you want to have a conversation later about this, we'd be love to talk to you about it and talk about things that we could do.
Sure, I can send you an email and can discuss about that. I have another questions, but we have no time, okay? And we can discuss by email. Thank you very much for your amazing
That'd be
talk.
wonderful. Thank you.
So thank you, Erik. We can talk later, and I have some questions, but I can write to you.
Yeah, sorry I went so long. Sorry.
No, it's our fault. It's because of the technical issues that we are just running. So thank you very much, Erik, and we can talk and discuss soon. Okay?
Yeah, that sounds wonderful. Thank you, everyone, for
Thank you very
attending
much.
and sticking through. Thank you.